Thank you all for having me today. Thank you for that introduction, Adam. Today I'm going to talk to you guys about how we're using AI to develop protein-based biotechnology. Since it is the IFML Year 5 celebratory event, it's going to be a lot of highlights over the work that we've done over the last few years. So before I start talking to you guys about proteins and AI, I want to talk to you about why this is very important and how AI for biology has become a pillar in AI research. A lot of this kind of started with Google getting involved in the protein folding problem with AlphaFold 1, then AlphaFold 2, and then AlphaFold getting spin-off from DeepMind into Isomorphic Labs. And Isomorphic Labs is now working on more protein AI for drug discovery applications. Around this time, Meta didn't want Google having all the fun, so Meta actually got really involved in also protein language models between 2019 to 2022, and they've even made their own little atlas where they folded 6 million proteins using their Meta AI ESM fold models. And then NVIDIA joined the party after that, providing this infrastructure and this cloud place called BioNeMo, and it's all for life science, machine learning, data science, deep learning. And since then, OpenAI has recently gotten into AI for biology. ByteDance actually has launched their own version of AlphaFold 3. They call it ProteinX. Fun fact, the lead developer on that project actually came from the Deep Proteins group. And Apple just launched a model called SimpleFold as of a month ago. So it's an area where all the major tech companies are embracing. And why are they embracing AI for bio? And it's because biotechnology can revolutionize so many fields, right? So if you look at the pre-AI biotech world, biologics were already bringing in close to $500 billion a year in revenue because protein-based drugs are very critical to our health. You can think of vaccines. So Jason McClellan, professor here at UT, he actually engineered the spike protein that went into the COVID-19 vaccines that came out in December 2020. AI is revolutionizing right now cancer research. There's a lot of AI models coming out in cancer applications when it comes to either protein development or more at a higher level like cells or metagenomics or DNA and RNA sequencing. But because we're applying AI to speed up the discovery and the development of these molecules, there's actually a lot of emerging biotechnology markets that really weren't accessible for proteins prior to using a lot of this new technology. So some of these are anti-aging. So you can see OpenAI has a collaboration with Retro Biosciences for discovering and engineering proteins that can hopefully reverse aging in stem cells. This is a recent NSF grant from earlier this year given to Novonesis. That's one of the leaders in enzyme engineering in the world. They are responsible for making detergents, dish soap, laundry, and that's all proteins, right? People don't realize that detergents is biotechnology, right? So they are actually got a grant to use AI and automation to make human milk oligosaccharides. So replacing simple baby formula with actually more similar the human breast milk. Here's one this is actually from my PhD. We got a Forbes article for engineering an enzyme with AI for plastic bioremediation. I'll show that later in the talk. And that enzyme's actually been further extended in a variety of ways. One way is Lululemon's using it to recycle PET plastic into new jackets and yoga pants. So you can go get recycled, enzyme recycled Lululemon pants that were used to be water bottles, which I think is pretty cool. And then a big area where proteins are going to change the world, in my opinion, is in biomanufacturing, right? Here's one from earlier this year where AI-engineered peptides boosted yields for biomanufacturing in E. coli. So now that I've kind of motivated why it's important to develop proteins and use AI to accelerate the development of novel biotechnology, I'm going to talk about how I structured this talk. So first, I'm going to talk to you guys about what are proteins, just to get, establish a floor of jargon and some of the terms we're going to use. Then I'm going to talk to you guys how we have developed AI frameworks for protein engineering, talk about some case studies, stability fine-tuning, and also what we call cross-modality fine-tuning techniques. And then we're going to jump into AI for protein design, and I will discuss the difference between protein engineering and protein design in this talk. And we'll talk about two frameworks, this ambient protein diffusion framework, and if we have time, we'll dive into this PRODIT framework. So to begin, first, what are proteins? Before we can even begin to discuss what are proteins, we have to understand what are amino acids. So amino acids are the building blocks of proteins, and they consist of having a backbone and a side chain. So if you see here on my screen, the backbone is everything that is not this R group, right? So this amine on the left and this carboxylic acid on the right, along with this carbon in the middle and this hydrogen, this is essentially the backbone of each amino acid. And what differentiates every amino acid from each other is the variable R. So biology uses 20 different R groups, and you can see they range from different properties. Some are positively charged, others are negatively charged, others are more greasy and like to, they're not very water soluble, and then a few have very unique properties. And it's actually how you combine these 20 amino acids, right? So our vocabulary is these 20, and it's how you actually build strings from these 20 amino acids that you actually get a protein. So you can see we start having a unique sequence of these amino acids, and based on the exact sequence that you're writing, you start developing what's called secondary structure. So that often looks like these little helices or these arrows that are coming together. So you have either beta sheets or alpha helices. And then whenever you take different secondary structure elements in the sequence and fold them into a 3D shape, that's when you actually get like the assembled protein. And this is often called the tertiary structure, right? So the sequence itself is primary. All of these secondary structure elements that you form, like these helices and beta sheets, is the secondary structures. And then bringing them all together is the tertiary. But often, for a lot of proteins to do their job, one protein can't do it by itself. So different proteins come together to build an even bigger complex, and that's known as the quaternary structure. So often whenever we're talking about proteins, depending on the exact function and application, it's either going to be like a monomer, which is its tertiary state, or it's some higher order complex, which is the quaternary state. And it's this 3D shape that they adopt, whether it's the tertiary or the quaternary structure, that allows a protein to execute its function. So for example, you can see this protein here, its 3D shape is to form a circle with a hole in the middle. And seeing this hole in the middle, you can probably guess what its function is, right? Its function is to allow molecules to pass through, kind of like how we cut cheese. And then here's actually one of my favorite proteins. This is ATP synthase, and you can see it spins, and its job is to produce energy from the food and the oxygen we eat, similar to how a turbine spins in a dam to produce electricity. So this key understanding that the structure of the protein is what actually endows it with its function, and all that is encoded in the sequence. So the whole goal of developing proteins or biotechnology is encoding function, right? So that you can do that primarily through protein engineering or protein design. So protein engineering involves when you take a natural protein and you improve its function by making mutations in it. And this got the Nobel Prize in 2018 for the directed evolution technique, and it's what's often used to date to engineer proteins and develop biotechnology. So you can see here are some examples of some proteins, and you can see the mutations that were made are shown either in pink or purple on this left one, or in pink on the right. And they're often just like a handful of mutations, 1, 3, 5, 10, 15. But most of the protein itself, which is on average like 300, 400 amino acids, it's still natural. While on protein design, it's actually the opposite. You tend to start with a function in mind, some like ambiguous function you want. Maybe you want it to eat plastic or cure triple negative breast cancer. Like just pick whatever function you want. And then from there, you work backwards and design a sequence from scratch that would hopefully fold into some 3D shape that can execute that function. So here, there was a Nature paper. This is like a new field, I would say. It's really kind of taken off in the last 3 to 5 years. Here's one of the initial Nature papers where they developed an AI model that the function here was to generate proteins that look like letters. And you can see they succeeded on making these computational proteins where it spells out the alphabet and the numbers. While this function itself of making letters is very not useful, you can see that in the future we can apply it to societal problems where we actually need a protein that has a specific function, like eating a specific type of plastic or cleaning the oceans with whatever toxin we're talking about. So as we dive into some ML models, I want to just establish some definitions or some terminology. So often you'll hear me use the word residue, and that has to do with the exact amino acid and its position in the string. So it's, the residue would be like at position 20, there is an arginine. So taking the position and the identity, that is a residue. And then whatever nature puts there, we call that wild type. And then whenever we want to mutate what nature has, those are our mutations. And the whole goal of mutating proteins is to change a phenotype. So phenotype is just another word for whatever function we're talking about in a protein. So first I'm going to talk to you guys about MuCompute. This is a work towards for my PhD, and it's essentially BERT-style self-supervised learning on protein structures. So the way it works is we're going to identify a residue that we want to focus on. You can see it here in the middle is the pink. We're going to select the neighboring atoms shown in green. We're going to filter that out, and then now we're going to delete the pink residue. And now we have our input, which is a surrounding chemistry, and we have our label, which is the residue that we deleted. So it's essentially just BERT-style training, but instead of using a language model, it's on a protein structure model. And the goal here is to train the model to identify residues where the wild-type amino acid is incongruent with its surrounding chemistry. So just putting all that together, how do we actually use this to engineer proteins? You can see here we have a protein. We run this algorithm on this valine 216. You can see the probability distribution is pretty uncertain, kind of even across 3 different amino acids. Lots of possibilities will probably work here, probably something we shouldn't focus on. Here is at tyrosine, at position 14, there's a tyrosine. You can see the model likes a similar amino acid to tyrosine being phenylalanine, but the chemistry of all of these 3 amino acids are very similar. So these would be considered conservative mutations, but probably not the thing that we're looking for. While, for example, here at glutamine 39, you can see the model actually prefers an arginine or a lysine, right? They make up 98% of the probability distribution. That has very different chemistry from a glutamine. So here, the model thinks fundamentally a different chemical entity belongs here than what nature put there. These are the ones that we look for. So you can see this glutamine 39, it's essentially what we're doing is we're trying to identify hotspots or anomalies to prioritize our protein engineering campaigns. And then whenever you want to visualize this across the structure, here's kind of what it would look like, where all the red locations are where the model strongly disagrees with what nature put there. And that's how we can isolate and identify the subset of the protein that we can target in the lab to actually make proteins and improve their phenotypes. So we've done this on various case studies. Here was our proof of concept protein back in 2018, 2019. So the wild type protein gave off blue light. You can see we improved its phenotype to give off blue light. It's 8x brighter. During the pandemic, we made a polymerase that allowed you to do isothermal COVID-19 diagnostics. We stabilized it so you can just use your stovetop rather than requiring fancy instruments. You can see here's our AI engineered protein still working. Everything else is dead. And we actually shipped this all over the world during the testing shortage summer of 2020. Here's a cancer therapeutic protein where we used AI to improve its expression. So this is the phenotype of interest. And this allowed them to finally make enough protein to begin doing animal trials with the protein. And you can see this. Here's an animal data where the rat is living much longer, or the tumor's growing much slower than if you didn't use this therapeutic enzyme. And then here's some on biomanufacturing applications. So we made some of the first few enzymes where you can actually use light to change its chemistry. We used our model to engineer it, one paper to use blue light, another paper to use red light. And then here is just a pure biomanufacturing play on an enzyme that modifies an amino acid for like raw materials. I'm going to go through one case study. Those are all high levels of other enzymes. So here's one that we find interesting, and we showed earlier, we had a Forbes article on, and this is when we engineered an enzyme to eat plastic. So this is called the PETase, and there was other ones that were being found that also had this phenotype that were called cutinases, and these two proteins are dramatically different in sequence. You can see they're about 300, 330 amino acids in length, and only 48% of them are the same. So over half the protein is very different between the two when you look at the sequence. So that's what, like 150, 160 mutations. But whenever you superimpose their structure, you can see they look very, very similar in 3D space. And this is the reaction that the enzyme does. So it takes polymers of PET and gives you the monomers TPA and ethylene glycol. So this was a hot topic, and it still is. And you know, a lot of labs were working on engineering these proteins. You can see the wild-type enzyme here in purple, and different mutations were found by different labs all over the world to improve the activity or its ability to eat plastic. But we ran our model on it, identified 3 mutations, stacked them together, and our enzyme was significantly better than anything else that was out there at the time. We named our enzyme fast PETase because it was much faster. And here is data where we went to Walmart, bought all this PET food packaging, and degraded it with our enzyme. So you can see this one took about a day and a half, while this one over here took about 4.5 days. And here's a time-lapse video of our enzyme just eating a cookie container that we bought at Walmart. So we bought this, we ate the cookies, and instead of throwing it away, we just fed it to our enzyme. So the next case study I find really interesting. This is how we combine these structure-based models with AlphaFold. So this is for API. API is an active pharmaceutical ingredient, API biomanufacturing. So there's this drug that is an FDA-approved pharmaceutical for Alzheimer's. It's called galantamine. And galantamine essentially is only found in daffodils, and specifically daffodils in like Scotland. So like if Scotland decides to not sell us their daffodils, we lose access to this drug. So why don't we just pull the proteins out of the daffodil, put them in a bioreactor, and just brew the drug like you would brew beer? That was like the goal of this project. So at the time of starting this project, we had just identified the first few enzymes that were in this pathway of making this FDA pharmaceutical. And that enzyme is the one that turns this molecule into this major product, 4-O-methyl-norbeldine. So all it's doing is at this oxygen right here, this OH, we're going to add 1 carbon to it. And we want to add it at this oxygen, not this other oxygen. This is a byproduct, and we don't want that. So my friend Simon, a very gifted protein engineer, currently now at Harvard. He does a lot of transcription factor biosensor engineering. He developed this very complicated genetic system to try to use green lights to identify enzyme variants that improve the function or production of this molecule. And you know, he tried techniques like randomly mutating the protein or combining, you know, different libraries to mutagenize the protein. And this didn't work, that didn't work, and you know, Simon was kind of feeling like this. And around this time, I had developed a new model, and I was like, hey, why don't we give it a shot? So our new model is Compute X, and the difference is it is now able to predict masked amino acids, but can also be conditioned on other molecules like vitamins and ligands, DNA and RNA. So, and this is around the time that AlphaFold first came out. So why don't we combine AlphaFold, other AI docking models, and our Compute X model to have a pure AI stack to engineer this protein, since traditional protein engineering techniques didn't work. So what we did was we AlphaFolded the protein, we copied over the SAM vitamin to the structure, then we AI docked the substrate to the protein, made a completely computational 3-molecule structure, used it to generate designs. We're going to screen those designs and then combine them together to make a better protein. So here's kind of what this computational structure we generated looks like. You can see here is the folded structure from AlphaFold, and then in green and in blue are the computationally docked molecules. So this is all on the computer. No, we have no idea if this is real or if it's not, but like, let's just give it a shot. So here are some of the designs we made. We only made 22. I think 7 of them improved activity. What is interesting is at this key position, which you can see right here, we ran the model with and without the vitamins and ligands present, and then we made the mutations that the model suggested with and without them present. So you can see here, the model predicts a methionine mutation when the ligands are present, and when it's absent, it predicts this arginine. So you can see, using this context of the ligand, gave us a successful mutation that was significantly successful, while excluding this context killed the enzyme. So you can see here's the performance of that key mutation. We ended up getting doubling the KCAT and almost by 3, almost cutting the KM, giving us over, I think like a 5x improvement, right? 3 divided by 0.64 on the enzyme's properties. So then we solved the structure. This is what it actually looks like after we engineered our protein. Here's our mutation shown in orange. And here's the before and after. So even though with this error, our model was still able to find mutations in the active site, even though the structure was wrong. And you can see here's kind of what we started with. Here's some other designs, and here's our stacked variant. And you can see we have about a 3 to 4x improvement on how quickly we can produce these pharmaceutical intermediates. So now that I've finished this case study, I'm going to transition into some of the cool AI models that we've built on from this point. So around this time is whenever deep proteins first started, and we were thinking a lot about this. This was the main problem, and we didn't know how to move forward. And that was that these BERT-style self-supervised learning models, we realized that that is not the appropriate pre-training task to learn good protein representations. Because as we trained the model, we moved over from CNNs and ResNets over to transformers and a graph transformer. You can see our model gets much, much better at the pre-training task of predicting what we masked. But the whole point of protein engineering is not to predict what you masked, but to predict mutations that improve the function. So there's this disconnect between the pre-training task and what you actually want to use the model in the lab for. And you can see as it gets better at the pre-training task, it actually gets worse at predicting experimental mutations. So what do we do, right? How do we iterate or make this model better in a self-supervised or unsupervised way that actually leads to improvements downstream in the wet lab? So at first, when we started this, we were like, okay, we're just going to take our pre-trained model, take the feature vector from the encoder, and kind of get rid of the classification head, and instead add a regression head to predict experimental properties that you can measure in the lab. Around this time, protein stability was the only phenotype that had a decent amount of data for us to experiment on how do you do downstream supervised fine-tuning. So we converted our structure model. We made the stability oracle model where we took mu compute and it can now fine-tune it on experimental data. And then at the same time, we took Facebook's language model, ESM2, and did something very similar where we would just grab the representations from the backbone and then add a regression head on top of it to predict it. So we have this, either a sequence model or a structure model, and we're training them on stability data. So when we started wrangling with the stability data, we realized that the data sets were very small, right? So we curated a much, much larger test set. We call it T2837. And then took the remaining data that existed publicly and filtered out anything that was similar to the test set to have a very, very different training set, which is the C2878. But what I wanted to point out here is you can see that this data is very small, right? Almost 3,000 mutations, almost 3,000 mutations. And we have about 380 mutation types, right? So that's essentially this 20 by 20 matrix, but subtracting the diagonal, because you can't mutate the self. And you can see the data is extremely biased. Most of the data you can see is mutating any amino acid just to alanine, which is why this column is super red. And we have a lot of mutation types where we have just no data, which is all these off-diagonal blues with no numbers in the middle. So we invented a data augmentation technique that we call thermodynamic permutations to try to address this. So in the literature at the time, what they would do to improve their data, they would just, instead of mutating from a wild type amino acid to the mutant with a corresponding label, DDG, they flip it. So you can mutate from the mutant back to the wild type and then just flip the sign of the label, which is great for doubling your data set size, but it doesn't fix other biases, which I'm going to show you here. But in thermodynamic permutations, what we realize is if you have the same position in the string, the same environment, and you have that amino acid, that wild-type amino acid mutated to multiple amino acids, so maybe like a leucine to an arginine, that same leucine to a lysine, that same leucine to a tryptophan, et cetera, et cetera, et cetera, you can now actually mutate in between those mutations. We call this thermodynamic permutations. So you can connect from mutant 1 to mutant 2 through the wild type and actually augment the data where you're mutating from mutation 1 to mutation 2, and you just take these two DDG signs and subtract them from each other to generate a thermodynamically valid label that was never actually computed or generated in the lab. So this allows us to grow our data set up to 10x, and it also balances the label of the data set, which I'll show here shortly. So here is applying thermodynamic permutations to our test set. So you can see very biased to alanine mutations. We generate from 200, almost 3,000 mutations, we generate an additional almost 8,000 mutations. Notice how there's no more blues in the off diagonals because we have now all mutation types covered, and the red has been attenuated, right? It's not as deep red as it is over here. So then combining the original data with thermodynamic reversibility, which is this one on the left, and thermodynamic permutations, which is this one in the middle, here's kind of our final data set where we go from almost 3,000 to over 13,000. And then so here's on the label side. So that's all on the input side. You can see the original data is orange, and it's all primarily to the right of this, meaning destabilizing or negative mutations. Whenever you flip it with thermodynamic reversibility, you get primarily positives over here in blue, while our technique gives you very balanced. It's evenly centered around negative and positive values. So very important to get to fix the class imbalance when training these models. And then so while we were developing this model, this big data set came out. They invented some new technique where they can essentially tie the cleavage of proteins to stability. It correlates pretty well. And it was about, almost 600,000 mutations. We curated a subset of this. We call it cDNA 117 to minimize any overlap with our training test set. And you can see it's still, even though it has all mutation types present, it still has biases. You can see this is slightly red, this is slightly blue, these are slightly red. So by applying thermodynamic permutations on it, or TP, we go from about 116,000 mutations to over 2 million, giving us a data set that's about 2.2 million to train a model, largest data set to train, to do supervised fine-tuning. So upon doing this, we train our language model, Prostat IFML, and our structure model, Stability Oracle. Here's on our curated test sets. And then on literature test sets, and you can see we get SOTA across the board, where our structure and our language model are essentially 1 and 2 across the board. Which was really exciting to see. We got this published in Nature Communications. And then around this time, we were also looking at, is it possible to take AlphaFold as a feature extractor and do a similar thing and have it predict stability of mutations? So that's where we came up with this mutate everything architecture, where you just run it through the AlphaFold backbone, and then we're able to expand the representations to get an L by 20. So L is the length of the protein, 20 is the 20 amino acids. And then we can just decode all these representations using either a single mutation head or higher order mutation head, shown here in blue. So whenever we focus on the single mutation head, you can see that mutate everything is SOTA. It matches or outbeats Stability Oracle on all these metrics: Pearson, Spearman, precision, recall, AUC. But the reason why we call it mutate everything is because while other methods require you to run, to make a prediction one at a time, because you do only one forward pass for the entire protein through the model and then decode just with a lightweight head, we get essentially flat run times to make, 10,000, 100,000 mutations. And I think we were able to push it to over 1 million, while other models scale linearly. And so here's the results of mutate everything on higher order mutations. You can see we hit the highest AUC and Pearson correlation compared to the literature models, and we published this in NeurIPS. So once we learned all of these like data wrangling, data biases, techniques to do supervised fine tuning, we were wondering how can we take what we learned and apply it to other phenotypes? Well, the problem with applying it to other phenotypes is there just aren't large experimental mutational data sets for like expression, KCAT, activity, even really binding affinity. Those are starting to be generated now, but there's still a long ways to go compared to that cDNA data set. So we were kind of stuck here, and we're like, hey, how do we leverage these things to do other things in proteins? Because stability isn't everything, right? Stability only gets you so far. Often other phenotypes are the problems to commercializing a protein. But then we realized, can we just turn to nature for this data? So before I dive into how we did this, to give you a little bit of background, there's this thing called multiple sequence alignments, or MSAs. And it's whenever you take your protein of interest and then you search massive database to find all of its brothers, sisters, cousins, uncles, grandparents, just how the protein has been evolving through time. And then you can now align on a per index what mutations does nature sample at this position over the course of like evolution. So you can see, for example, like these mutations are highly conserved, you know, nothing has been mutated, while some of these other ones, you can see it likes one or another amino acid, doesn't really try many, while other ones have a variety of mutations, kind of like here. So, but the key thing about an MSA, it's not just what nature mutates things to that we observe, but it's also the absence of observance. What do we not see it mutated to? So if you often don't see any mutations at a specific position, it's because it's most likely destabilizing or it kills the function, so nature prunes it. While other mutations either improve stability or function or are just neutral. They don't really have an effect. So we think of an MSA of how evolution has been mutating proteins, and we thought, can we distill how evolution allows mutations based on a protein structure? So we went back to our mute-compute architecture, and we then have this microenvironment here centered on a residue. So you can see this residue here is this one that we select. We now look at the distribution on this column, which we plot here on the right. And we can now compute what we call an EVO score, where you can take the probability of observing this amino acid that you want. So you pick whichever one you want. Maybe we want to mutate this histidine and to this alanine. Right, so you can see we can take the histidine's probability of 0.25 and the alanine of 0.06, and then get the log-odd ratio of the two. And then this is what we're going to train the model to learn, this EVO score. And again, we're just using the stability oracle architecture, but just swapping out the label from experimental DDG data to an evolutionary score. And doing this, we see that we no longer overfit to predicting wild type. Right, it kind of plateaus around 0.6-ish. But we keep getting better at predicting experimental results of what someone would see in the lab. So our EVO rank loss is, this objective is aligned with engineering proteins in the lab. And so here we can see we evaluated on a variety of different benchmarks. Some of them are for binding affinity, some of them are for protein stability. And the previous generation model, mute computes in green, and then our new mute rank model is in blue. And we have two other, like David Baker's Protein NPNN and Facebook's ESM2. And the thing about our two models, they're trained on the exact same data set and the same architecture, we just changed the loss. And you can see, we see significant improvements from green to blue, both on regression and classification tasks for binding affinity and also for stability. Next, when we compare this to other self-supervised models across a variety of test sets, you can see there's a big gap between the blue and the rest of them. The rest of them are kind of all clustered together because the ceiling for them is their wild-type masking self-supervised loss. But if you look at our blue, whenever you take these three models down here, the green, orange, and red, and then fine-tune them on experimental data, just like how we did with Mu Computer Stability Oracle, you can see that they all get better. But our mute rank model is competitive with models trained on experimental data, even though we didn't use any experimental data. So we recently took our stability oracle model and our mute rank model and used them to validate them in the lab, where we made mRNA biomanufacturing enzyme for mRNA vaccines. We improved it about 13 folds. And then here we show where we compare our previous model, mute compute with mute rank, where we get significant zero-shot performance improvements, but we also get significant improvements when doing the supervised fine-tuning to build stability oracle. So you can see we're able to match the performance of stability oracle v1 with about 10x less experimental data. So next we wanted to apply our evil rank loss to protein language models, right? We did it in a structure, can we do it now in a sequence? So when we first tried this, we realized that because protein language models are trained on evolutionary data or these sequences via BERT-style training, we saw no improvements when you switching it over to this evil rank loss. So we kind of had to go back to the drawing board. We didn't really know what to do. It was very, we were very confused. So then we took a step back and kind of examined really what was going on that made the evil rank or the mute rank model work. And what we realized was it was actually this cross-modality self-supervised fine-tuning that was the key to evil rank. So what I mean by that is in mute rank, our input is a mass structure, and then we're now training it to predict this evil score. So this is structure input, sequence output, this cross-modality between in the self-supervised task. So what does that look like for a language model, right? For a language model, the input is a mass sequence. So I guess that means we've got to come up with some structure score. So what would be a good structure score? What is a structure score? So that's when we came up with this technique called structure tuning. So structure tuning is essentially the inverse of evil rank. So instead of fine-tuning, doing self-supervised fine-tuning of a structure model with evolutionary data, we're doing self-supervised fine-tuning of a language model with structure data. And the way it works is we pass, the structure through an encoder. Now we get an embedding. We then discretize or tokenize that embedding into a code book, and then use these structure tokens to supervise the language model. So we tried 3 different structure-based models for our structure tokens. Fold-seq is a backbone only, so it doesn't look at the side chains or the identities of the actual amino acids. It's just looking at the coarse-grained structure. While our mute computer, mute rank architecture is an all-atom masked microenvironment, so similar to fold-seq, but it has all the atoms. And ESM3 does just a global structure tokenization scheme. So we took our entire training set, used these 3 models to essentially come up with structure tokens. And you can see that here's our model that we started with, ESM2. Here is the literature baseline where they actually have a model that takes as input the sequence and the structure. So our model's input is sequence only. And you can see when we use fold-seq, we get improvements over ESM. ESM3's tokenizer, structure tokens, actually made performance worse. And then using our mute rank or evil rank representations actually gave us the best performance. And it nearly matches with if you take a language model and explicitly give it the structure as input. So that's why we call this model ISM, because the structure is implicit. It's the implicit structure model. And you can see here, whenever we take the representations and we also do supervised fine-tuning on our stability data set like I showed earlier, you can see we get significant performance improvements, not as good as our mutate everything or our stability oracle, but much, much better than our previous one with Prostato. So now I'm transitioning to the last highlight of my talk when we talk about generative models. So no longer protein engineering frameworks, but protein design. Okay, so I'm gonna fly through this pretty quickly here. But essentially, de novo protein structure backbone generation with diffusion models has become a pretty hot topic over the last few years. So essentially, you just start off with a 3D Gaussian of atoms and then run diffusion model on it to generate 3D structures. Here's an example of what this looks like whenever you're conditioning it on this peptide. And voila, it makes a structure from nothing. And David Baker actually got the Nobel Prize for this last year. So what does the workflow look like in protein design for these de novo protein models? So you use a diffusion model to generate the backbone. We use this MPNN inverse folding model to generate sequences that will fold into that backbone. And then you do a variety of different computational filtering techniques, and then whatever passes that is what you would move forward to experimental validation. So how does this workflow look like in more detail? So you generate the backbone, inverse fold it to get a sequence. Now you want to forward fold it, and then you get all these metrics out. The main metric we're going to use is designability, meaning if you align the backbone and the forward folded sequence, how well do they align in 3D space? Do they have under 2 angstrom error? And then you take this subset of highly designable protein sequences, and then you cluster them. And you see how many clusters you get for your designable subset to measure how diverse are the proteins that we're making. So that's our diversity metric. So designability and diversity kind of trade off between the two. And what we ended up doing here is we came up with this technique called ambient protein diffusion. And the motivation behind it was that these diffusion models cannot generalize to larger proteins, right? The average length of a protein is 300 amino acids, and you can see that the performance starts dropping significantly as you start approaching this average. And we also realized that most of the data that is used for these models are thrown away, so everything to the left of this is not used because it's considered low quality data. And here's kind of what this low quality data looks like. You can see like in blue, a lot of it is folded, but then you have these like noodles that kind of connect them, and then you lose like that whole tertiary interaction. Like, so for example, is this little folded motif here correctly oriented in 3D space? Like, should it be here? Should it be over here? Should it be over here? We don't know because the protein is poorly folded. So currently, all these models, just rather than trying to leverage the information in this data, they just throw it away. So we started looking at how do we use ambient diffusion, which my good friend Giannis invented during his PhD, to be able to train with corrupted or partially corrupted data. So we started looking at these low-quality proteins as corrupted data, and what we came up with was we can just use them at different diffusion time points. So if it is a poorly folded protein like this, we would only use it at very high noise times, while if it's a pretty well-folded protein like this, we'll use it for more time, and if it's a high-quality protein, then we will use it for the entire diffusion time. And using this technique of like where you only partially use a protein at different T's during diffusion allowed us to significantly improve the model. So you can see here's our baseline model that we started with in blue, and then we applied it, we applied our ambient technique, got it much better, and then we fine-tuned it on larger proteins, and you can see it even improves it more. But around the time when we were developing this, NVIDIA decided to get into the protein design space, and they launched this model, Proteina, and their model is gargantuous, 400 million parameters. Ours is like 20 million parameters, trained on 21 million proteins. I think we're training on, like 500,000 proteins. So rather than going with a large scale, we realized we just have to improve the data. So what we ended up doing was we realized that our protein has a lot of duplicates in them. You can see here's 2 proteins in the training set where they essentially look the same. It's just this little tail here. AlphaFold did a poor job folding, similar to this tail here where AlphaFold did a poor job folding. This is really just duplication in the data set. So we came up with this pipeline to clean up the data set, going from roughly 2.3 million to about 300,000. And this is the statistics of that data set. And then applying ambient protein diffusion to train on it, we have a Pareto frontier model when it comes to short protein, where we're beating all the Proteina models. And then on the long eval, you can see our model has the best designability and diversity when comparing it against Proteina here in light blue. Which is cool to see that, we here at UT, we were able to beat NVIDIA beat NVIDIA on a protein design task. So I would like to thank you all for coming to my talk, and also thank everybody that was involved in this work. We had a handful of computational scientists, experimental scientists, and this is very interdisciplinary research, so a lot of different faculties from different departments that contributed here. I'd like to take any questions.
Kristen is getting set up. Can you unplug your laptop? We can take a question for Danny.
Yeah. So, has there been any work on, quote unquote, multimodality of like, there being 2 modalities, one of which is, let's say, proteins or structure, and like something else, maybe biological or maybe language?
That's a very good question. So walking over here, I actually cut out like the next project, which was a multimodality diffusion model where it co-designs the protein sequence using discrete diffusion and then the structure using continuous diffusion. And we've shown that you can condition this model on protein function, where it will start generating sequences and structures that resemble how nature will execute this function by placing the correct amino acids in 3D space and relative to each other in 3D space. But we're currently working on further evaluating that model. Is that it?
Any other questions? Okay, thanks, Danny.